Yeah. Okay. And I saw Kelvin and Kelvin was gone. Just like so at the bottom. And then I put my Kelvin this weekend. I said,
All right. Good morning. How's everyone doing? Monday. It's week 3. Yeah. Talk about mental. I know. Weather's terrible. All right. All right. So, let's see. Today we are going to continue moving through Mendel, and I think we should get close to finishing up this lecture so we can move into chapter 4, which is extensions of Mendel and non-Mendelian inheritance patterns, which is kind of where things get a little bit more complex for most people. So we're going to try and do that. You have another worksheet due Friday. Have people been looking at it at least? Okay, good. All right, so start working on that. And then we have lab this week. I'm sure there's an exam coming up at some point. I think the week after next is our first exam. So yeah. Questions on anything so far that I talked about last week? Oh, I should mention, I did put in your pre-assessment grades. They were great. That makes me feel okay. The class average was somewhere in the 35-ish percentile. This is a pre-assessment. You already have 30% of what you need. You're doing good. Okay, so I originally had it in there as a grade that is currently counting, but then everybody was failing the class because it's like the only grade in there. So it is present. You should be able to see it, but it should also say excluded right now. Here's the deal. If you do better, again, on the final knowledge acquisition assessment, I will replace that grade. If you don't, it stays as is, and I will release that column like towards the end of the semester so you can see how that figures into your grade. But for now, I didn't want people to, be like super sad that they're failing genetics before they've even gotten a chance to show me their true skills. So, I think the high was like a 50%. I think it was 27 out of 50 was the high, so just over 50%. So, take heart. It'll be fine. Okay. Now that we've got that out of the way, let's learn some genetics so that those grades go up at the end of the semester. Okay, so we finished covering monohybrid crosses with Mendel, right, which is just crosses that are involving or looking at one particular trait. I did just want to quickly recap this. So I'm going to start here by looking at all of the monohybrid crosses that Mendel examined, showing sort of the outcomes, right? So the F1 generation, we can use this column to tell us what, or to verify what concept. Okay, so we're crossing our parental strands, which are our true breeders. F1, all of them have a particular trait, right? So all of the F1 offspring are either tall or dwarf. They're all tall. They're all round or wrinkled. They're all round. So what does the F1 phenotype tell us about these two traits? Right, so using the F1 generation, we can determine which of these traits is dominant. Okay? So that's the important information that we get from F1. F2, these are the results that Mendel counted, and again, he had pretty big sample sizes. We also not only want to look at sort of the raw number of tall to dwarf or round or wrinkled individuals, but we want to put that into a ratio. Okay, and so this ratio, close to 3-to-1 for most of these. What does that tell us? What principle does this validate or verify or support. Yes. So this 3-to-1 ratio using a monohybrid cross validates or supports Mendel's first law of inheritance, which is the law of segregation. So for every one of the traits that Mendel studied, these two things were true. The F1 generation showed just one parental phenotype, and the F2 generation came out in a roughly 3-to-1 ratio for those two variants or those two phenotypes. If you think back to those early theories of inheritance, one of them was blending inheritance, which means that offspring are the intermediate between parents, right? So if you have a tall parent and a short parent, you'd be sort of exactly in the middle. This tells us, well, even this, our F1 and F2 generations tell us that that cannot be possible. So we can automatically say that whoever came up with that idea had not looked at parents before. Okay, so we already talked about that. I'll kind of skip over these. All right, and so this is, again, just showing us what Mendel did experimentally and sort of our outcome. This is sort of a nice representation because it has the parental plants with their traits. It shows us their genotypes. From the genotypes, we can determine what gametes are made. We can imagine these two gametes fertilizing, or these two gametes fertilizing, or those two gametes fertilizing, and giving us our F1. Our F1 generation, I think I cut it off, is all big T, little t. Okay, oh no, here it is, sorry, big T, little t. Again, we can imagine segregation here, and then self-crossing with all of our F2 generation phenotypes. We're going to sort of see this idea of how gametes merge, fertilize, come together multiple times, and so I always think of this as foiling, just like in math, right? So when you're trying to figure out what combinations of gametes have when they're lined up like this, you can, first outer interlast it and then end up with what's down here. We'll see this again in a minute. But I always think that. Okay. So using this concept, we can sort of bring this back to what we just talked about with meiosis. Right? At the time of Mendel, he didn't know about chromosomes. So that's why I remember we called them like, what were they called? We called them gemmules, I think. But you didn't call them alleles, they're like factors, because he didn't know what it was. So we can kind of take what Mendel was thinking, put it together with what we learned about meiosis, and then we can come up with what this actually looks like. And this is just using one chromosome. Okay, so we start with one parental set of chromosomes here. We all know that this is probably what we envision the outcome to be. Right? 2 mom, 2 dad, and our gametes, 50-50 sort of split. Keep in mind that we can have crossing over. And in this case, just because of the locus or the alleles that we're looking at here, these gametes actually have the same big R, little r, little r. But crossing over in other cases can change the genetic composition of the chromosomes. So this is just a table from our textbook that shows you what happens or what your outcomes are if you have parents with particular genotypes. And you could work through these on your own, hopefully, using a Punnett square. But what generation have we talked about that would look like this? So this is one individual, we cross it with another individual. Which of our generations so far has been heterozygotes? F1, right? So this is the outcome that we would expect from an F1 cross, which is two heterozygotes cross, or an individual is heterozygote self-fertilizing. We designate the genotypes like this. So 3 out of the 4 are going to have a big A and then something else. Remember that we're looking at phenotypes most of the time. If I showed you a tall pea plant and asked you what phenotype it is, could you tell me? If I ask you what genotype it is, could you tell me? Right, because what are the options? Right, big A, or big A, little a. You can't tell the difference because the plant looks the same. So 3 out of the 4 have a big A. We don't know what the second allele is all the time. The other quarter, we can tell because only little a individuals are short. This is one that we'll talk about more coming up here. This is called a test cross. Well, actually anything where we have an unknown individual and we cross it with a homozygote recessive individual is a test cross. But in this case, if we have a heterozygote and we actually back crossed it to one of our parents, so to a homozygote recessive parent, we would get a 1-to-1 ratio. If you have 2 homozygote dominant, all offspring are homozygote dominant, 2 homozygote recessive, all of them would be homozygote recessive. Here, this is our parental lineage, right? All of them come out heterozygotes. And then here, not a parental lineage, but also everybody comes out homozygote, or comes out dominant in phenotype. All right, so you can kind of refer back to this if you want. On the other hand, you'll probably end up knowing most of these by the end of this class. If you want to improve those pre-assessment scores, you'll end up knowing these by the end of class. Or you can figure them out by hand, right? These are fairly simple. And there's more. All right. Any questions on our law of segregation? Each gamete gets 1 allele for every trait. Everybody's good with that? Excellent. Okay, so now we are going to go to Mendel's sort of next experimental step, which was looking at dihybrid crosses. Dihybrid crosses become a little bit more complex because now we are looking at 2 traits for each parent, or 2 traits in each plant. So an example is we may cross a plant that has round peas that are also yellow with one that has wrinkled peas that are also green. So that is the difference between just looking at round versus wrinkled or just looking at yellow versus green to now looking at the combination of these 2 things. So what we are trying to differentiate or determine using this type of cross is whether traits are linked when they are sorted into gametes or whether they assort independently. So here are the 2 hypothesized possibilities for how chromosomes or genes assort. So on the left, we have linked assortment. We're going to walk through this carefully. If we look at the parental generation, we have round yellow individuals. Okay, so this parent is round and yellow, right? This parent is green and wrinkled. What are the potential what are the potential gametes that come from this round yellow seed? So what alleles will be present in the gametes?
Big R, big Y.
Yeah, so it can be big R and big Y, and that's it. Okay, so in this parent, we are now assuming that we're dealing with 2 chromosomes. 1 chromosome has our R alleles, 1 chromosome has the Y alleles. Okay, and so if we go through meiosis with this, can everybody do that? How many people are comfortable or confident that they can take this cell and draw out meiosis? Yes? No? Somewhere in the middle? This is how I feel when I tell my daughter to put her dishes in the sink every night. Why? Okay. I'm just kidding. It's not that bad. 5-year-olds aren't infuriating anymore. Okay, so what is the first thing? What did I just do? I also can't draw it out, so don't feel bad. What is the first thing that we would do? What? No? Yeah, it's terrible eraser. What do we do? What is our first step in meiosis? Somebody please tell me. Replication! Okay. Thank you. So when we replicate, oh, don't know what that was. We are going to have this. You guys are going to get so tired of drawing this stuff. Is that right? No, that is not right, Brant. Keep doing. Is that right? Okay. What happens next? Yeah. All right, so that's the next thing we'll draw is metaphase. On our metaphase plate, over here and here. Yeah? Okay, you can tell me if I'm wrong. What's next?
Separate.
Okay, how do they separate? Yes, that is the correct mechanistic separation, but what do they look like in the next cell?
There's a pair of chromosomes, R in one, and then a pair of the R in the other, and then Y in one, Y in the other.
Yes, so what do we call these things? How would we say? Oh, I guess I should draw two. So what? Use your words. Nope. What composition of chromosomes is here and what is here? What do we call the pairs or the sets or the? Yeah, sister chromatids. So here we'll have, how do I do this? And here we'll have this. Remember that we're still in our sister chromatid pairs. Right? Okay, and then what's our next step look like? Metaphase, so fortunately I drew these so that we can just metaphase them out. And then what? The sister chromatids separate, right? So here is where we get our gametes, right? So we get two here, and two here, and two here, and two here. And so we get one, 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 one. One, one, one. This is R, R, Y, Y. Okay. Well, thank you. This is tough. Okay. So, what is the allelic composition of every single one of those gametes? Everybody together. What alleles are in those gametes? Big R, big Y. We just did this. Every single one of those gametes is big R, big Y. This parent only has big R's. This parent only has big Y's. Every gamete is going to have one copy of each of those. We just talked about that with segregation. Big R's, big Y's. So if we did this same thing with our little round wrinkled guy, what do we get in those gametes? Little r, little y. It's all that parent has to give. Okay? When we cross a big R, big Y gamete with a little r, little y gamete, remember we were cross-fertilizing our F1s, or our parental generations, what does our F1 look like? Okay, yeah, we get a big R from one parent and a little r from the other. We get a big Y and a little y. So every F1 is big R, little y, big R, little r, big Y, little r. It's the same exact thing for both hypotheses so far. Okay, so the next step is where it changes. With linked assortment, what we are assuming is that the chromosomes you got from one parent stick together when you're forming your gametes. Okay? Because they're linked. So if you get a big R chromosome from one parent and a big Y chromosome from that same parent, linkage or linked assortment would tell us that that big R and big Y stick together when you're forming gametes, and so you get a gamete that is big R, big Y. Okay? So everybody still with me? Very good. So in linked assortment, we have only chromosomes in the F1 that look like our parental chromosomes, or our gametes. Our gametes look like our parental gametes. In independent assortment, we say that the chromosomes that we got from our parents, or the genes that we got from our parents, don't link. It doesn't matter who they came from. When you're forming your gametes, you'll get all new combinations. Okay, so we would say this is where we would FOIL. Yeah, look, it reappeared. Big R, big Y. Oh, it's red. Big R, little y, little r, big Y, and little r, big Y. Right? So we're saying that we can have any of those four combinations. As long as you get one R allele and one Y allele, your gametes are good to go. Okay. So, Mendel, oh, now I can't erase. Why? Why is this happening to me? Alright, let me do this. Let me do this. Okay, well there's going to be big red marks all over our screen this entire time. Awesome.
Computer and mouse. You can click the mouse thing.
This thing?
Yeah, and then like there.
Glad you guys know what I mean.
This one I want to. The shrine is still there.
Oh, God, Jesus. Alright! Okay, so I can get that part gone, but not the rest of it. That's awesome. And class is over. Just kidding. Do that. Maybe if I can do it. Hold on. I just repeat the same things over and over. Don't actually work right. Alright. Alright. Oh,
Try and use the mouse here too. Like,
this
use that and try and click on buttons up there.
yeah, oh.
Try it. You might have to use your PowerPoint annotation.
Oh, I
So,
see. This.
that, the little pen. Erase.
Oh, thank you. Fantastic. Okay. Now that I got that figured out. I know, it's great. Okay. All right. So what Mendel does next is test this theory, not this theory. The theory of linked versus independent assortment. Okay. Now this is going to look a lot like what we just talked about because we start with parental generation. Our parental generation are true breeders, and what does it mean when we're true breeders?
Homozygous.
We're all homozygous dominant, okay? And so we start with one parent that is homozygous dominant for yellow round seeds and one parent that is homozygous for wrinkled green seeds, and we cross-fertilize. Okay, so we remove the ability of these plants to self-fertilize, we cross-fertilize, we end up with an F1 generation that looks like this, round and yellow. And then we allow that F1 generation to self-fertilize. So in self-fertilization, we end up with gametes that look like this. Does this look like independent assortment or linked assortment? This looks independent, so let's take a look at what our F2 generation looks like. So we're going to work it out. Yeah. Oh, it's not in there. I'm making you write it. Okay, so for dihybrid crosses, we use Punnett squares just like for monohybrid crosses. What is symbolized across the top? One parent's what? Gametes, right? These are gametes, right? We have our little spermies up here, and we have our little eggs down this side. If you always think about these in gametes, hopefully it makes sense. Do you want to see the most common mistake I get when people are drawing out dihybrid crosses? So you know when you're drawing these out, if you do this, I will be sad. Makes it sound like I'm sad a lot. So the most common mistake I see is this. And then the board goes funky. What's wrong with that? Okay. Segregation tells us that every gamete gets one copy of each gene. How many copies of the R gene did you make this gamete have? 2. Yeah, so not right. This messes up the first law of inheritance. Okay? So we always need to make sure that our gametes have one copy of one gene and one copy of the other gene, and not 2 copies of any of the genes. Okay, so when you're doing this dihybrid cross, this is where you would foil. I'll tell you what your parents look like. So I'd say your parents are heterozygote for round and wrinkled, or round and yellow seeds. So you would know that they look like this. So to get the first gamete up there, you do your first. To get the second gamete, well, not quite in the same order, but you do your outer, inner, and last. And that would give you big R, big Y, big R, little R, big Y, little R, little y, which is different order than it is up here, but the same gametes. It doesn't matter what order it is across the top or down the side. Okay, so if you're going to fill these in, what are you going to put in the upper left-hand corner? All big. Big R, big Y, big Y. Next. Should make you guys go down the row and tell me next. Yeah, there we go. Big R, big Y, little y. Next. Yeah. So everybody's furiously writing, so I'm assuming that you're doing these as you go. So for a dihybrid cross, you should always have 16 squares. And using your genotypes, you should be able to determine the phenotypes. So up here, the phenotypes are shown with actual pictures of the peas. But you will also need to be able to figure out what the, because when you're doing this, I don't think is anybody drawing peas, sprinkled, yellow, green, brown? No, okay. You're just writing the genotypes. You should be able to tell me then what this genotype is going to give you, right? Big R, big Y, going to give you round and yellow. Okay, so let's just look at this for a second. From here, what is your phenotypic ratio of offspring? So let's start here. What are our potential what are our potential offspring phenotype?
Yellow and round.
Okay. So we can have, yellow wrinkled, yellow, round. Gross. So we can use a board that doesn't work, or I can use a whiteboard that doesn't erase. It's a win-win.
Oh, the options.
No. All right, so we have yellow round, and I wonder why it takes me so long to do this. I heard yellow wrinkled, green round, and green wrinkled. Okay. So these are our potential offspring phenotypes. What is the ratio of these? So how many of those 16 are yellow and round? 9? How many of them are yellow and wrinkled? 3. Green and round? 3. And green and wrinkled? 1. Perfect. So we would say that we have a 9 to 3 to 3 to 1 ratio of offspring. Again, this is phenotype. I will just go ahead and tell you that I'm not going to ask you for a genotypic ratio in your dihybrid cross because you have like 8 members. There's a lot of different genotypes here. So if you say 9 to 3 to 3 to 1 ratio, you should always be talking about a dihybrid cross between 2 heterozygous parents. Phenotype outcome of the offspring. Okay. Okay, and so this is, again, sort of the whole thing all in one. We have what the plants look like here, and then we have what we're talking about when we're talking about gametes and genotypes and phenotypes on this side. I don't think I have a lot of extra to say here, so I'll just leave it as when you need to go back and look at what Mendel did and look at it at a couple different levels, this is a good place to look. Okay. So just like we looked at monohybrid cross data from Mendel, this is Mendel's dihybrid cross data for this particular combination. Does this give us a 9 to 3 to 3 to 1 ratio? Somebody do math. Looks like it, and yes, are not necessarily the same answer. How would we determine if this is a 9 to 3 to 3 to 1 ratio? Okay. You could count them all up.
You divide them all by 32.
You divide them all by 32 is the best way to do it, or the fastest way to do it. You could divide them all up. If you were going to test statistically if these were actually in a 9 to 3 to 3 to 1 ratio, you would add them up. But the easiest way to just look at the ratio we have here is divide everything by 32. Right? So if you divide 32 by 32, you get what? If you divide 108 by 32, you get 3 point something. I know y'all have phones.
Roughly 3.1.
Roughly 3 would earn you no points on an exam.
3.15.
3.8-ish. 3 point, so if you divide 101 by 3? 3.2. And if you divide 9.8. Okay, 9 point, there we go. All right, so this is the actual experimental ratio that Mendel got. Here in a few minutes, we should be able to decide if this is actually close to 9 to 3 to 3 to 1, or if there is a statistical difference between what he got and what our expected output is. Okay. I feel like everybody's angry at me today. Okay. It's all right. So if we go back and looked at linked assortment, what we would actually expect in our F1 generation would be half of the individuals to be yellow and round, and half of the individuals to be green and wrinkled. So does Mendel's outcome of 9.8 to 3.2 to 3.4 to 1, nope, 3.8 to 1, closer to 1 to 1 or closer to 9 to 3 to 3 to 1? Yeah, obviously much closer to 9 to 3 to 3 to 1. So we can sort of go ahead and assume that, or say that this hypothesis of linked assortment is not supported by this particular experiment. Can anybody think of a scenario in which we might see this? When might we see traits that appear linked? Yeah.
Wouldn't it be if like the specific trait is either like one or the other and it's just a one?
I'm not sure I follow.
Like if you're only like looking at like tall or short.
Oh, so if you're only looking at, yeah, so if you're just looking at one, so if you're just looking at round versus wrinkled, it would look 50-50. But when we're looking at two traits, when might we expect them to be in a 50-50 ratio in the offspring? I made an assumption, but I didn't say out loud that everybody just went with.
Wouldn't it be that if it's a dominant thing?
How many chromosomes did we use going through this? One for each trait?
So if they were on all the same chromosome?
Yeah, so if both of these traits, so if the trait that coded for coloration and the trait that coded for key and shape were on the same chromosome, is it really easy to like break a chromosome in half? And it's possible, right? We do have crossing over. But would we see a 9 to 3 to 3 to 1 ratio if they were on the same chromosome? It would be much closer to 50-50. All right. Okay, so this is what this actually looks like if we write it out. This is a different way to represent what happened. All right, so we have at the top our 4 possible male gametes, which were what we had at the top of our Punnett square. The next row is our female gametes, which is what we have along the side of our Punnett square, and then those are the 16 potential combinations. So if you really are bored of Punnett squares, you could try writing it out this way and see how it goes for you. All right, and then down at the bottom, this is the statement for the law of independent assortment. So as gametes form, we get segregation of any pair of hereditary determinants. What the heck does that mean? Remember, we don't have...
Maybe like what we know as chromosomes?
Right, so this would be what we know as chromosomes. We have segregation of any pair of chromosomes independent of other pairs of chromosomes. Right, so just because you make gametes that have chromosome 1 from mom doesn't mean it's also going to have chromosome 2 from mom, and 3 from mom, and 4 from mom, and 5 from mom. It's going to be a random assortment, which is, when we go back to this, this is independent assortment, right? So the fact that we make 8 million plus gametes just from independent assortment. Okay. Okay. Okay, so I'm gonna walk you through another Punnett square. This time we're gonna look at a test cross instead of part of Mendel's experiment, so we're not doing parental or F1. We're going to say that we are starting with an individual that is phenotypically dominant, okay, so its phenotype is dominant, and we want to know whether it's homozygote dominant or heterozygote, because we can't tell those two apart. The easiest way to determine that is going to be by crossing that individual with a recessive individual. So if we cross it with a recessive, we can then look at the offspring that come from that cross and determine if the other parent was homozygote dominant or heterozygote. So if we cross this individual with this individual, what are their offspring going to look like? This would be a tall plant with yellow seeds crossed with a short plant with green seeds. What is the F1 going to look like, or what is the offspring of these guys going to look like? Yeah, so every single one of their offspring would have this genotype, and so they would all be tall and yellow. If we cross this plant with a homozygote recessive individual, what do the offspring look like now? Are they all this? No. So what we actually end up with is a 1 to 1 to 1 to 1 ratio. So it would be tall yellow, short yellow, tall green, short green. Okay, so using this individual here and here, you can determine the zygosity of the other parents. So is it a homozygote dominant or is it heterozygote by looking at the outcomes? Okay, so we're going to actually walk through an example of this. You are starting with, I'm just going to show you currently. Nope, I don't know what happened. Okay, let's go ahead and check this one to see if we get the right answer. I'm going to try this again because I'm a glutton. We're going to cross this guy with this guy. Okay, so when you're writing your Punnett square, because we're crossing two, or we're looking at two traits, we should have 16 boxes. Wow. Oh, is that? Alright. Alright, so what goes across the top?
Big T, little T, big Y.
Okay, big T, big Y.
Big T, little Y.
Big T, little y.
Little T, big Y.
Little T, big Y.
Little T, little
Little T, little y. Okay. What goes down the side?
Little T, little Y, all the way down.
All right. Ooh. Yeah, I got a little sketchy there at the end. That's okay. So what goes here?
Big T, little T, big Y, little Y.
Big T. What? Little T. Big Y. Big Y. Little y. Next.
Big T, little T, little
Okay.
Y. Little T, big Y, little Y.
Okay. And
All
I
the
did something wrong.
All the way.
There we go. This is a big Y.
all of them.
All of those. What? Now it's like randomly erasing things. Okay. So this is what our top line looks like. Because each of these three is identical, the rest of this is going to fill in exactly, right? so these would all be the same, these would all be the same, these would all be the same, and these would all be the same. So when we're looking at phenotypes, what is our phenotypic ratio? So this one is going to look what?
Big and yellow.
Okay, so we're going to have 4 that look big and yellow, tall and yellow,
Tall and green.
4 that look tall and green, 4 that look short and yellow, and 4 that look short and green, which gives us our 1 to 1 to 1. Alright, so hopefully everybody got that, right? Okay, so we can do Punnett squares with more than 2 genes. This is what it would look like with 3. As long as you remember that across the top and down the side are gametes, you should actually be able to do these without too much of a struggle. Alright, but this is, how many squares? 8 times 8, 64. If I gave you 5, you'd be looking at 16 by 16. So, it just gets a lot.
Are you gonna make us do this?
No. I haven't yet, but I'd have to give you a lot of time. Okay, so this is again taking us back to the idea of combining Mendel's law with what we know about chromosomes now. We've already seen this, so I'm just going to leave it here for a second and remind you that this validates or supports Mendel's law of independent assortment. Hopefully none of this is shocking to us. I'm not going to teach this because it's annoying. Alright, so I am going to take the next minute and a half and just start talking a little bit about how we know if what we got was different than what we expected. Has anybody done a chi-square test yet? Yes. Okay, good. We have some people who have done this. We're going to do a lot of chi-square in this class. We're going to start now. We're just going to keep going all semester. Hopefully by the end of the semester we can figure out what we're doing. Alright, so what we want to know is if we go back to this idea of 9 to 3 to 3 to 1, right? This is expected data. This is what we figure out without any actual living organisms involved. We can all do the Punnett square. We can all imagine the chromosomes segregating, making gametes. When we take gametes and make them into offspring, do we always get the perfect expected ratio of 9 to 3 to 3 to 1? No, we just saw that with Mendel. He got 9.8 to 3.2 to 3.8 to 1. The question becomes is what Mendel got close enough to that to actually be saying the same thing, or is there some other factor that's influencing the segregation and the gamete formation? Okay. So, starting on Wednesday, we will walk through how to do chi-square. This is the formula. We have our observed and our expected values, and that's all we need to figure out if there is a statistically significant difference between our expected data and our experimental data. All right. And then once we do that, we will talk about exceptions. Okay, you should have enough information now to be starting on that worksheet. By Wednesday, you'll have enough to finish it up. I don't think I put very many chi-square on there. Let me know if you have questions. I will see you all in lab tomorrow. We'll be practicing this. Have a good Monday. Oh my God. I miss freshman. What was it? It was the scientific method. Because that's how all the things were.
Oh my God. I wish.
I wish we could go back
I
to
was
that.
like I was.
Huh? I know, right? I was thinking the same thing.
How's it going? Oh,
Good. Thank you.
I saw you got an elk.
I
did. Oh, did I send it to you?
No, Dr. Berger.
Oh, I did?
Yeah, he's like,
Yeah.
this part is huge. And I was like, that is a big heart.
Yeah.
A lot of pain.
Yeah, 170 pounds. Yeah.
I was talking and I was like, would you actually say that or am I making that up? Anything philosophy?
No, I think it's pretty good. Like just the.
No.
Yeah.
It's
How
actually
about,
easy.
Okay.
A lot easier than like
I
the.
think it like actually works though.
Just from 8 to 10.
No, you and me.
Yeah.
Let's just talk about it then.
It's not
And I
like
can kind of walk you through
Yeah.
exactly what it was.
Okay.
Yeah, so we'll just, that's fine. It should be all like dry lab stuff. And so we'll just have to turn it on the next station.
Alright, sounds good. Thank you.
Yes.
I think I should bring it up.
I think I did at least. Did you look at yours?
Sorry.
No, you're good.